The goal of this project is to assess how the androgenesis and dioecious sexual systems manifest in Australian Selenium hybrids across generations, especially relative to the parent's role as pollen donor or pollen recipient. The dioecious sexual system is relatively rare in angiosperms, occurring in only 6 to 10% of them. The Selenium genus is particularly useful for understanding sexual system evolution, because not only does this genus display the rare dioecious sexual system, but also the androgenesis and the hermaphroditic. Figure 1 displays the androgenesis sexual system on hybrid flowers. You can see a single inflorescence containing small, staminate flowers with a larger hermaphroditic flower at the base. On the other hand, the dioecious sexual system displays full separation of staminate and pistulate flowers on separate plants, sometimes with the presence of functionally pistulate flowers due to inaperturate pollen. In 2018, Daniel Hayes, a previous undergraduate researcher at Bucknell University, crossed 18 species of Solanum. We chose Solanum dioicum and Solanum ultraspinosum as the parent species, because not only does Solanum dioicum display the dioecious sexual system, and Solanum ultraspinosum display the androgenesis sexual system, but they're also morphometrically distinct from each other, which makes changes due to hybridization more apparent in the hybrids. While all crosses with Solanum dioicum as the pollen recipient failed to produce viable seed, crosses where Solanum ultraspinosum was the pollen recipient resulted in high rates of success. In figures 2 through 4, all diagrams labeled A show the pollen donor morphology, while B shows the pollen recipient's morphology, and C showed the F1 hybrid morphology. Our findings suggest that the F1 hybrids are most influenced by the pollen recipient species, and as such, all hybrids display the androgenesis sexual system. However, when these characters were analyzed using PCA, we found that while the hybrids were most similar to the pollen recipient as expected, the hybrids were distinct clusters, suggesting that they are distinct hybrids. The same measurements and analyses will be conducted on the F2 hybrids, which are currently growing, and further generations. We are specifically interested in changes in floral architecture and the absence or presence of an aperturate pollen, which could be indicative of a transition to the dioecious sexual system.